Welcome to this lesson on the Fluid Mosaic Model. This groundbreaking concept revolutionized our understanding of how cell membranes are structured and function. The Fluid Mosaic Model describes the cell membrane as a dynamic, flexible structure rather than a rigid barrier. It explains how the membrane maintains the integrity of cells while allowing essential processes like nutrient transport and cell communication to occur. The Fluid Mosaic Model was proposed in 1972 by scientists Singer and Nicholson. Their work revolutionized our understanding of membrane structure. The core concept of this model is that cell membranes are not static or rigid. Instead, they are dynamic and flexible structures where proteins float freely within a lipid bilayer, much like icebergs floating in the ocean. This flexibility is crucial for the membrane to perform its many functions. The name Fluid Mosaic Model comes from two key characteristics. First, the term fluid refers to the fact that the components of the membrane can move laterally within the membrane plane. The lipids and proteins are not fixed in place, but can slide past each other, giving the membrane its flexible nature. Second, the term mosaic describes the diverse array of proteins embedded in the lipid bilayer. These proteins are arranged like tiles in a mosaic, creating a varied and complex pattern across the membrane surface. The cell membrane consists of four main components. The phospholipid bilayer forms the fundamental structure, with hydrophilic heads facing outward toward the watery environments inside and outside the cell. Membrane proteins are embedded in or attached to this bilayer, performing various cellular functions. Cholesterol molecules are interspersed within the bilayer, regulating membrane fluidity and stability. Finally, carbohydrates are attached to proteins and lipids on the outer surface, playing important roles in cell recognition and communication. Membrane proteins fall into two main categories. Integral proteins are permanently embedded in the lipid bilayer and often span completely across the membrane from one side to the other. These transmembrane proteins are firmly anchored and play crucial roles in transport and signaling. Peripheral proteins, on the other hand, are temporarily attached to the membrane surface or to integral proteins. They are not embedded in the lipid bilayer and can be easily removed without disrupting the membrane structure. Membrane proteins serve four essential functions. Transport proteins, including channels and carriers, move substances across the membrane that cannot pass through the lipid bilayer on their own. Signal reception proteins act as receptors that detect external chemical signals and trigger appropriate cellular responses. Cell recognition proteins, particularly glycoproteins, identify cells as self or foreign, enabling the immune system to function properly. Finally, some membrane proteins have enzymatic activity, catalyzing important biochemical reactions right at the membrane surface. Three main factors affect how fluid or flexible the membrane is. Temperature plays a significant role. Higher temperatures increase membrane fluidity by making lipids move faster, while lower temperatures decrease fluidity and can make membranes more rigid. Cholesterol content is also important because cholesterol molecules maintain optimal fluidity by preventing extremes in membrane flexibility. They keep membranes fluid at low temperatures and stable at high temperatures. Finally, fatty acid saturation matters because unsaturated fatty acids have kinked tails that prevent tight packing, thereby increasing membrane fluidity. The Fluid Mosaic Model fundamentally transformed our understanding of cellular membranes. Before this model, membranes were viewed as static barriers that simply separated the inside of cells from the outside environment. This model revealed that membranes are actually dynamic, functional structures essential for life. The key insight is that membrane flexibility enables vital cellular processes including cell signaling, nutrient transport, waste removal, and cellular communication. Understanding this model helps us comprehend how cells interact with their environment and maintain the delicate balance necessary for life.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.